DNA is the hereditary material found in almost all organisms. Nearly every cell in an animal's body contains DNA. When located in the cell nucleus, it is called nuclear DNA, and it encodes the genes found on the chromosomes. However, a much simpler form of DNA can be found in the semi-autonomous structures called mitochondria that are responsible for cellular energy metabolism. Unlike nuclear DNA, many copies of mitochondrial DNA exist in each cell. In all DNA, information is stored as a code generated from 4 complementary chemical bases: adenine, abbreviated as A, guanine, G, cytosine, C, and thymine, T. Like nuclear DNA, sequences of these 4 bases constitutes genetic information, represented as a string of letters. Different species have unique forms of mitochondrial DNA, and we can identify them from samples of biological materials, including blood, hair, scats, and other tissues. The polymerase chain reaction, abbreviated as PCR, is a laboratory technique that produces multiple copies of a DNA sequence by amplification from extremely small sample sizes. A basic PCR requires several components, including the DNA template to be amplified and primers. The primers are a short, single-stranded DNA molecule that binds by complementarity, where guanine binds to cytosine and adenine binds to thymine on either ends of the DNA segment to be amplified. The PCR amplified product that may produce billions of copies of the target region can then be subjected to further testing. For instance, the amplified fragments of DNA can be separated by gel electrophoresis. In this technique, DNA molecules are separated in a medium or a gel by an electrical field. The PCR will produce multiple copies of the target region DNA if the primers are complementary to the template DNA in the primer binding sites. However, the PCR will not produce copies of the target region DNA if the primers are too different and not complementary with the template DNA in the primer binding sites. Realistically, however, the PCR can still produce copies of the target region if the primers have very few differences in complementarity compared to the template DNA in the primer binding site. The capacity of a primer to bind to the template DNA and originate a positive PCR amplification depends upon the number of base pair mismatches. Mismatches at the 3' end of a primer make the primer less likely to anneal to the template than those mismatches near the 5' end of a primer. This is influenced by the methods used in the laboratory. During the PCR, the temperature of the sample is repeatedly raised and lowered through 3 steps, called denaturation, annealing, and elongation, to assist the DNA polymerase copy the target DNA sequence. The binding of the primers on either ends of the target segment occurs during the annealing step, usually performed at between 50 to 65 degrees centigrade. The capacity of a primer to bind the template DNA and originate a positive PCR amplification depends upon the annealing temperature used. The lower the annealing temperature, the higher is the probability of non-specific binding of the primer to the template DNA. A species-specific PCR can be simply described as a pair of primers that are designed only to originate a positive PCR amplification in the presence of DNA from a single species. It is a test specific for one species only. So, in order to avoid a false positive result, the primers cannot anneal to the DNA of other species. In other words, the objectives of a species-specific PCR are twofold. Firstly, to produce copies only of the target region whenever DNA from the target species is present in the sample, and not to produce copies of a target region in the presence of DNA from another species. In an accurate species-specific PCR assay, the primers anneal to the target species DNA. However, the primers do not anneal to the DNA of other species that might be present in the same sample. Using gel electrophoresis in the laboratory, this is how the results of an accurate species-specific PCR might appear. The PCR product from the two samples containing the target DNA appears as a solid dark band on the left-hand side of the gel. These samples are consequently determined to be positive. On the right-hand side, as no PCR product was amplified from the other 7 samples that potentially contain DNA of other species, these samples are determined to be negative. In the case of an inaccurate species-specific PCR assay, the primers may anneal to the target species DNA. However, the primers also anneal to the DNA of other species that might also be present in the sample. Therefore, the primers are not specific, and false positives, or type 1 error, occurs. Using gel electrophoresis in the laboratory to assess the PCR product arising from a flawed test, the false positives are indistinguishable from true positives arising from the target species DNA. A Fox-specific PCR assay was designed by Berry and others and used in the Fox eradication program. According to the authors, they developed a highly accurate and reliable DNA-based PCR multiplex test that identified foxes from field-collected faeces. Primers were designed to amplify a 134 base pair PCR product in the presence of fox DNA only. Problems with the Fox-specific PCR assay were later noted by the same authors. Of 186 scats that yielded positive results using this test, 47 scats were found to contain fox-like sequences, but could not be conclusively identified as those arising from foxes. Much of the non-fox amplified product was thought to be DNA derived from rabbits or hares. We sought to better evaluate the specificity of the PCR assay developed by Berry and others against DNA from a wider range of Tasmanian species, including some that occurred in the diet of endemic Tasmanian predators. In our experiment, DNA samples were harvested from blood, muscle, kidney, liver or spleen and were collected from 20 different species, including the red fox that was taken from Australian mainland populations. The DNA was extracted from the samples according to standard protocols. We tested the putatively fox-specific pair of PCR primers designed by Berry. The primers were predicted to amplify a 132 nucleotide region of the cytochrome B gene of the red fox. We performed PCR amplifications with the putatively fox-specific F and R primers using different annealing temperatures either by gradient or conventional PCR. The results were that foxes and several other species yielded positive amplifications at different annealing temperatures. In this example, because DNA from the hare is amplified at 62 degrees Celsius, non-specific DNA synthesis occurs at even higher annealing temperatures than those previously reported in 2 studies by Berry and Saar. The PCR produced clear amplifications for the red fox, European rabbit, and European hare, as well as domestic cattle and domestic pigs. PCR products were also obtained for the Tasmanian devil and long-nosed potteroo with weaker intensity. At an annealing temperature of 56 degrees Celsius, strong amplifications were obtained for several species, with the exception of the long-nosed potteroo. A single cattle, rabbit, Iberian hare, and both pig samples yielded a larger quantity of amplified PCR product than either fox samples, suggesting that in some cases the primers designed by Berry in 2007 amplified non-fox DNA more effectively. Increasing the annealing temperature to 60 degrees Celsius only slightly decreased the intensity of unspecific PCR products compared to the lower annealing temperature of 58 degrees Celsius originally used by Berry and reported in 2007. At 60 degrees, DNA from rabbit, hare, cattle and pig was readily amplified. Overall, we found there was an insufficient number of primer template mismatches required to prevent amplification from a range of species that could be represented in scat samples collected in Tasmania. Significantly, in the F primer, only a single mismatch was detected in the terminal 11 position of the primer for the rabbit, potteroo and cattle, with other species having 2 mismatches. In addition, the 9 3' positions of the R primer had no mismatches in the rabbit and Tasmanian devil, 2 in the potteroo and only a single mismatch in remaining species. Overall, most mismatches were located in the central and 5' regions of both primers, with the terminal 2 bases at the 3' end being the same in all species. We concluded that even in the presence of high-quality DNA samples, the putatively fox-specific assay, developed by Berry and others and reported in 2007, amplified DNA from a range of widely distributed exotic, domestic and native species that included domestic cattle and domestic pigs. The putatively fox-specific primers F and R did not specifically hybridize to fox DNA, despite being designed with the intention to specifically exclude DNA that would be expected to be present in scats produced by a range of Tasmanian predators. Scats have multiple sources of potential contamination. Scats of omnivores in particular, with a highly varied diet such as foxes, quolls, bandicoots, as well as scavengers such as the Tasmanian devil, contain a complex mixture of partially degraded biological materials. Even if obvious prey contents are separated in the scat prior to analysis, the possibility of contamination throughout the fecal material must be assumed. The species-specific assay proposed by Berry and others must contend with the likelihood of strong PCR amplification arising from extremely abundant forms of non-fox DNA. European rabbits are particularly common in the diet of feral cats and foxes, and are common as roadkill that is known to be scavenged as food resources by marsupial carnivores such as Tasmanian devils and quolls. Common domestic livestock species appear to constitute an even greater risk of sample contamination from multiple sources. Meat carryover in human feces as sewerage, as well as waste from kitchens, abattoirs and food manufacturing processes, are a potentially significant environmental source of cattle and pig DNA. Commercial dog foods may also contain a wide range of cattle meat and byproducts such as hooves and horns, and both cattle and pig products are common components of fox baits. As a consequence, this diagnostic assay alone cannot be relied upon to detect red foxes because of the high likelihood of false positives. Irrespective of what post-PCR laboratory methods are used, primers F and R do not permit sufficient specificity for the unequivocal determination of fox DNA. Because wildlife management decisions may be increasingly influenced by data generated from molecular technologies, there is a need to adopt best practice guidelines, especially under conditions when the detection of a unique invasive species incursion may be considered sufficient justification to initiate a costly eradication program.